Welcome to ACS Webinars, bringing you the best and brightest minds in chemistry, live every Thursday at 2 p.m. Eastern. To view our upcoming schedule, please visit acs.org/acswebinars.
Professor Fortunak is a well-recognized expert and works with organizations including the World Health Organization, UNITAID, the Clinton HIV/AIDS Foundation, and the Kilimanjaro School of Pharmacy. Professor Fortunak's work activities and research activities include new chemical synthesis of drugs for the treatment of malaria, diabetes, HIV/AIDS, all utilizing synthetic reagents and pathways based on the principles of green chemistry. And with that, again, I warmly welcome Joe. The floor is now yours.
David, thank you very much for your kind invitation to speak and for the chance to present today. I'd like to thank the ACS for letting me make this presentation. So you've seen the title to what my talk today is going to be about. And I put a lot of slides in here that I want to get through in about 20 minutes. So there's some things that I won't read to you, but let me summarize what I'd like to get across to you today. An awful lot of the world actually lacks access to modern medicines to one degree or another. Green chemistry is actually making a dramatic contribution to decreasing the cost of critical drugs for the treatment of HIV/AIDS and malaria in low and middle income countries. And I'm going to be using that acronym LMICs to represent that. But one thing is that in order to make medicines accessible, scientists can't work alone. We need to work with political will and muscle. We need to work with drug manufacturers. And we need to work with donor agencies who provide money to make it available, to make medicines available in low and middle income countries. And some of those agencies include the Clinton Health Access Initiative, known as CHI. In fact, I have to single them out because they're one agency that has been particularly effective at recognizing that people need to be engaged across a spectrum of activities in order to enable something to happen that's really dynamic in the world. The Global Fund for AIDS, Tuberculosis, and Malaria, GFATM, is another one because they provide an awful lot of money for low and middle income country access to medicines. The U.S. PEPFAR, the President's Emergency Plan for AIDS Relief, is another. And UNITAID is a last organization that I'm going to single out. UNITAID is really spectacular in what they do because they actually have a few $100 million a year to spend, and they target it towards the introduction of dramatic new treatments that can affect fundamentally health of people in low and middle income countries worldwide. Now, something that I do that is much less associated with donor agencies is to teach good manufacturing practice and detecting counterfeit medicines, and to teach drug development and drug regulation in Africa in order to affect economic development in the countries there so that eventually donor agencies won't be needed for these purposes. Well, let me get started. I'm sitting at my office right now at my desk at Howard University in Washington, D.C. This picture was taken from the roof of the chemistry building. Howard is something that's a little bit different from what many of your experiences may be. It's an HBCU. We're a historically black university. We were founded in 1867 to teach freed slaves, and historically we average training about 20% of the African American PhDs that are turned out in the United States, and we also turn out quite a number of African PhDs who are in their country and black PhDs in chemistry worldwide. But what I'd like to use as a touchstone here is one of my favorite photographs. This is Olduvai Gorge. This is in the Great Rift Valley that runs up the eastern coast of Africa, and this is where we find the earliest evidence of what it is that distinguishes mankind as a species from other animals. Now, there are a lot of people who are registered for the seminar, and in fact, I'd love to get a shout out to Lakeland College, where some people on the summer research experience who've tuned in for this practice. For this presentation. But look, this to me, a lot of people having registered for this presentation, some of you got up this morning and you said to yourself, I'm a child of God. I have a personal relationship with him or her, and that's what drives my life. At the other end of the spectrum, some of you woke up this morning and you may not have thought about it, but you may look at this and say that we are a fluke of the universe and we don't have any right to be here, as an old song goes. But one of the distinguishing species of mankind is that all of us have been able to come together as a civilized society to make tremendous advances in the way that we live on this planet. And I'd like to highlight 3 of those for this talk today. And some of the major advances in decreasing mortality and improving the quality of life in civilized societies include the ability to access clean water and sanitation, and the ability to implement advances in agriculture so that we've got food security and distribution. And a third one is medicines. So I'm going to focus on medicines, and I'll start off with a few quick facts. Now, about 80% of us that are tuned in today actually come from the United States. Quite a few of us are foreign and from places as far away as Guyana that I see. But at the time of the U.S. Civil War, 1861 to 1865, about 1 out of 5 children born in the United States survived to adulthood. Putting a slightly more careful circumscription on this, in the year 1900, infectious diseases in the United States that are limited to influenza, pneumonia, diphtheria, kidney infections and diarrhea and intestinal infections killed about as many people as the death rate from all causes in the United States in 2009. Now, how that indicates to us what the impact of medicines are on a civilized society is, I've been to Nigeria several times. I like it there. I'm very proud of the fact that I have a certificate from the Nigerian Institute of Pharmaceutical R&D that indicates that I'm an honorary Nigerian citizen, and I consider it a very highly civilized country. But life expectancy there is about the same as it was in the United States in 1900. That's 48 years. And life expectancy in the United States today is about 78 years. And largely the difference, I would argue, between those two, and the difference, this argument is well supported by epidemiological evidence, is the lack of a high rate of access to medicines. So on the one hand, our current industrialized societies, us living in the United States or Hong Kong or France, is unsustainable for future generations. Terry Collins at Carnegie Mellon likes to point that out to us as one of the great practitioners of green chemistry, but I'd like for us to look at it in a slightly different, turned around perspective. And this is a picture of 2 people. This is the highlands of Tanzania, driving up from Olduvai Gorge on towards the Serengeti Desert in Kenya. The person on the left-hand side was 18 at the time, 3 years ago when I took that picture. It's my son. He goes to the University of Maryland now. The person on the right was 9 years old. He's a Maasai tribesman. He lives in a village. He sleeps on a mud floor, often maybe with goats. He attends school sporadically, and he spends an awful lot of his life herding cattle. So looking at these 2 people, it highlights the fact that access to opportunities and access to the fruits of technology is not equal, even having an industrialized global society that we have today. And my feeling about green chemistry is that we can say to ourselves as a group that the world is green when sustainable access, and by sustainable I mean for future generations, to the fruits of technology is more equitable globally for everyone. Well, turning that back towards the idea of medicines, the World Health Organization has a list of roughly 300 medicinal products called the Essential Medicines List, and it represents a minimum standard of access to medicines. So this would represent access to drugs for malaria and tuberculosis, but not, for instance, to access to any really kind of modern drugs for the treatment of diabetes or congestive heart failure or cancer. Modern medicines are pretty much completely unavailable for about 2 billion of the world's 7 billion people, somewhere around 30% of the world. The statistics coming from the WHO in 2009. But if we look at this in the sense of medicine's distribution, much the same way that we might talk about income distribution, the richest 15% of the world's population, so that's the top 1 billion people, consume 91% of medicines. The other 6 billion people consume the remaining 9%. That's not a real good standard, but there's something, an issue that's unspoken here that I would like to actually address. Drug discovery companies are where I worked for 21 years. It was an honor and privilege to work in drug discovery companies, but we cannot expect them selling important new medicines to the richest 1 billion people in the world to provide access for the other 6 billion people. That's not a model that works in any kind of a private enterprise system. In order to have sustainable access to medicines, what we need to address are the issues of the lowest possible cost of production and access, and production technologies that are sustainable for the long term. One needs to observe all applicable intellectual property, and that means patents, and medicines have to be of assured quality. The same quality for people in Africa has to be the quality that's given in the United States. And access to medicines has got to go beyond the highly publicly visible programs that deal with HIV/AIDS, TB, and malaria. Well, we pursue 2 different strategies for access to medicines. One of them is that I and the partners and the non-governmental organizations that I work with and that exist outside of me work with suppliers and with countries to identify the needs and manage the supply chain, and we try and invent new green chemistry, and all of this rolls up to try and secure the lowest possible cost of access to medicines for low and middle income countries. But again, I'm going to come to that second strategy that's largely unspoken to by other people in the world, is that we need to improve regional capacity to manufacture and regulate medicines. And what that moves into is to assuring the quality, to increase the supply of medicines available, and to expand access into diseases beyond HIV and malaria and tuberculosis. Because there are more people who die in Nigeria from diabetes and from high blood pressure than from these 3 other highly visible diseases. The people who supply medicines for low and middle income countries largely are generic companies in India and China. Now, I'm going to make a distinction between 2 different things here. One of them is called an active pharmaceutical ingredient, or an API. That's the drug and the drug alone. It's made largely by chemical synthesis or biological synthesis or from natural product sources. Drugs need to be formulated into a delivery form called an FPP, or a finished pharmaceutical product. Global pharma is still a significant player in supplying medicines access to developing countries, particularly in malaria. And another thing that we need to take into account is that although this isn't totally true, a lot of Chinese products are not quality assured, and this speaks to the regulation of medicines within China. But China is very largely a driver of pharmaceutical intermediates for making APIs and FPPs for the rest of the world. Now, to speak to green chemistry, I can't help but mention Paul Anastas and John Warner and touch on the 12 principles of green chemistry. So these were published back in the 1990s, and I've rolled them down into just a one overhead slide right here. And I've highlighted a few of these that are most applicable to access to medicines. And so we're talking here largely about organic chemistry and multi-step organic synthesis when we speak about manufacturing active pharmaceutical ingredients. And just to speak to these, we need to emphasize a few of these. One of them is that preventing waste is better than generating waste and treating it after the fact. And that actually rolls into the second one that I've highlighted in red, is that chemical processes need to maximize the utilization of materials that go into them. We need to avoid auxiliary substances and unnecessary steps in a chemical synthesis. And looking at number 6 there, energy is also part of processing and making medicines, so we need to minimize energy use. Catalysts are superior to stoichiometric agents. Even if stoichiometric agents can be recycled, that's a lot of mass that goes into green chemical processing. And chemistry processes need to be designed to minimize the potential for accidents, particularly in resource-poor settings. So when it comes to green pharmaceutical manufacturing, a few points I'd like to make are largely, look at these 4 here. One is that whenever possible in a chemical synthesis, manufacturing steps in which we break old chemical bonds and make new ones to make a new molecule must be combined into a single pot whenever possible. And there is something that Roger Sheldon emphasizes called an E factor. So if we think about making 1 kilo of a drug, the E factor is a measure of the amount of waste that we generated in terms of kilos per kilo of product made. So the lower the E factor is, the more efficient a chemical synthesis is. We need to choose materials to minimize impact to the environment when speaking about manufacturing in low and middle income countries. And in minimizing impact, we need to speak to both minimizing the environmental footprint of manufacturing and the capital or the investment footprint, because that is particularly a difficult hurdle to get over in terms of increasing regional manufacturing for medicines. But basically, this all comes down to actually being a good scientist and to understanding not only chemistry but engineering as well. So streamlining processes to reduce waste and cost equates to new green chemistry, and one very important way that needs to be implemented in order to increase the sustainability of operations is to look at going beyond typical chemical stirred tank reactors that turn out products in a batch-wise fashion, and to look at continuous processing through flow chemistry. Well, look at these drugs right here. These are the 6 drugs that are key for the next 5 years or so in providing access to medicines globally for the treatment of HIV/AIDS. Each one of these drugs is important in their own way. HIV/AIDS is treated for patients who are treatment naive with a triple combination cocktail of drugs. So for instance, in the developing world, in South Africa, an incredibly heavily used product is a combination of Efavirenz plus lamivudine plus tenofovir disoproxil fumarate. Another very, very heavily used triple combination in the developing world is nevirapine plus lamivudine plus zidovudine. But these 6 drugs together account for well over 95% of the medicines that will be used to treat HIV/AIDS in the developing world over the next 5 years. Here are some key drugs that are used for the treatment of malaria. Artemisinin is a natural product. The use for the treatment of malaria of artemisinin was discovered by the Chinese in the 1970s. One artemisinin semi-synthetically derived component, as artemether or artesunate or dihydroartemisinin, is combined with a synthetic drug to come up with combinations that are known as ACTs or Artemisinin Combination Therapies. So for instance, artemether very rapidly clears malarial parasites from circulation in the bloodstream, but it has a duration of action of only a few hours. Artemether is combined with lumefantrine, a synthetic drug that you see just below it, and lumefantrine has a half-life of as long as a week or even 2 weeks in human tissues, so that artemether clears the parasites rapidly. Lumefantrine accounts for a very sustained clearance of parasites, so that patients who are taking ACTs don't suffer from a relapse of the emergence of parasites known as recrudescence. So I would like to show you some examples of what green chemistry has contributed to, and here are 2 HIV drugs as an example. TDF is tenofovir disoproxil fumarate. It's cost in 2011 from generic companies in India for the treatment of HIV/AIDS, and it's purchased on very large scale by the Global Fund and other agencies, is about 35% of what it cost in 2006. And I can't tell you exactly what the percentage is, but I can tell you that new chemistry and green chemistry has contributed very significantly to this reduction. Another example is Efavirenz. The demand for Efavirenz for the treatment of HIV in developing countries last year was over 500 metric tons. EFV's cost from India in 2011 is 12% of what it cost in 2005. And what I'd like to actually do is to speak to this specific drug right now. And look at the synthesis outline right here. So the synthesis begins from 4-chloroaniline. And I'd like to show you simple chemistry that doesn't change. It's directed orthometallation. Wonderful chemistry developed very significantly by Victor Stikas, Queen's University in Ontario, is implemented and followed by this asymmetric addition step. This is where the action is that we want to focus on. And the question is this PROT, protecting group in red, and it speaks to the green chemistry principles. I'd like to focus on addressing only what's inside the box and how we get from the cost in 2006 to 2011, and it's 4 different generations of green chemistry improvements. So in a first generation of synthesis, this was actually a very good synthesis, but it employed a protection and deprotection scheme for that nitrogen. And alkyl lithium asymmetric addition was really very heavily contributed to by a wonderful chemist up at Cornell, David Collum. But the E factor for this overall synthesis was about 55 kilos of waste for 1 kilo of drug. In the second generation, a dialkyl zinc asymmetric addition was employed, but this was stoichiometric dialkyl zinc that was used. So there was no protection or deprotection of nitrogen needed, and the E factor is about 35, and that was a very, very substantial improvement. But a third generation synthesis has been recently published by people at Lonza and by Eric Carrera at the ATH in Switzerland, and this is being practiced now by Lonza and soon by people in South Africa at the company Pelchem that uses substoichiometric dialkyl zinc for this step. That's an improvement. I don't know exactly what the E factor is there. It's probably around 25. But a fourth generation synthesis is available in which catalytic inorganic zinc-2 salts are used for the asymmetric addition, and the E factor here is approximately 12. So I'd like to show you what's disclosed in the U.S. patent that came from the Shanghai Institute of Organic Chemistry that speaks to this. You'll see that in this green asymmetric addition step, we're using about 5 mol% of what's now an inexpensive zinc-2 catalyst, zinc triflate, with an asymmetric catalyst. You notice down below, we've got 2 chiral centers in this chiral ligand that's used in the amount of 2 mol%. This chiral ligand is actually very inexpensive because it's derived from the synthesis of chloramphenicol that is a broad-spectrum antibiotic that is still very widely used in the developing world. Now, I want to speak to classical process chemistry development in green chemistry as well. I've got a picture here of Adrian Williams, who's been a graduate student with me. I think that's Niagara Falls in the background with him, on his honeymoon that he went recently. But this is classical chemistry of process development with a lot of contributors that come from many different places. So this is the step right here. Look at the starting material over on the upper left. That's actually the active drug that we need to get inside cells, and it's bisphosphorylated to become an HIV drug. Down on the bottom right is what we need as a prodrug salt in order to deliver it to people so that it can be both dissolved and absorbed to arrive at the site of action for people who have HIV. But what I'm going to show you now is a high-pressure liquid chromatography printout of the reaction itself after 5 hours of progress. So the challenge here is look at what's in red and focus on that. The monoester is an intermediate in the reaction. We need to go from a phosphanic acid to make a monoester, then a bisester. In addition to maximizing the conversion of the starting material to tenofovir disoproxil, and the peak for that is highlighted in blue, we also need to minimize the presence of impurities 1, 2, 3, and 4 that are highlighted in red. Now, what I've shown below here on this slide is the actual identity of these impurities that show up in the TDF Stage 3 process. And focus on for a minute, what we're doing is that the NH2 that hangs off of the adenine ring in TDF is reactive under the process conditions. And we need to balance that with driving to complete conversion the production of the bisphosphonate ester. Well, we've published with quite a number of authors in the Organic Process R&D Journal procedures for doing this. And some of the keys for getting there are to use high-pressure liquid chromatography mass spec and HPLC in combination with NMR to identify those impurities and to also identify the kinetics of their formation and how to optimize the reaction itself. We found that some important things are to minimize water in the reaction, to use HPLC as an in-process control so that we can stop the reaction as soon as it's done because the product is rather unstable under the reaction conditions, and to use a phase transfer catalyst, a tetraalkyl ammonium bromide salt that improves the chemical yield, it improves the stability of the product, and it maximizes product recovery. Well, what is the future of people who need treatment for HIV? The number of people who need access to medicines and LMICs is going to go from 14 million in 2011 to 23 million in 2015. And this is in a world where we have a global economic downturn. The Global Fund actually canceled their 11th round of funding mechanisms in November 2011 because high-income countries who donate heavily to the Global Fund are not able to meet even their existing commitments, let alone to think about huge new ones. But what I want to show you here is a graph that comes from the Clinton Foundation. The last year for which complete statistics are available is 2010. But look at 2011. The best estimate shows that about 7.8 million of those 14 million people who needed treatment in 2011 are actually accessing HIV drugs. If the target, the WHO has set a target of reaching 15 million people on treatment by 2011, I'm sorry, by 2015 out of that 23 million who will need it. So we've done a lot. Look at how few people had access in 2003, but there's an awful lot more to do. What I want to show you for comparison is look at the drugs over in the column 2011 volumes in metric tons. So TDF that we've been speaking about, 200 metric tons of it was used in the developing world to treat HIV in 2011. If we want to treat 15 million patients, though, given the distribution of what they should take, 884 tons is needed. And if we want to treat all of the 23 million patients in the developing world who need it, the real need is 1,771. For Efavirenz, you can see current supply for the developing world 500, and the need for 23 million patients is over 2,000 metric tons. So I see you get the point of this. But one other thing I want to speak to is we do work on industrial development in Africa, and the question is, why would you do this? I do this with Steve Byrne from Purdue University. So I'm a chemist. He's also a physical organic chemist, but his specialty is in the solid state properties of drug molecules. And with Sister Zita Ekiocha, who actually has 3 degrees in pharmacy at the St. Luke Foundation Kilimanjaro School of Pharmacy. This is in Moshi, Tanzania. It's very near Mount Kilimanjaro. The partners who work in this program are the St. Luke Foundation, which is represented by the Kilimanjaro School of Pharmacy, and their industrial pharmacy training unit. So we have Purdue and Howard University, and we teach 4 courses in a sequence, and we teach African pharmaceutical professionals and national drug regulatory agents in how to manufacture drugs and how to assure their quality, and this is supported by the United Nations Industrial Development Organization. So we teach 2-week-long courses to professionals who pay their own money to get there, and we teach them about 10 hours a day, 5.5 days a week, and they take exams when they're done. And the idea is that we can teach them how to make their own medicines and how to assure the quality of those medicines, because there's something very important. It's regional production can help with effectiveness and sustainability. If you're a donor agency, if you're the Global Fund, how would you feel about giving money forever simply to purchase medicines for Africa, for HIV? Donors expect that the reason they're donating funds is that there will be progress that comes out of it, so that eventually the person who is the recipient doesn't need donation anymore. In addition to this, we teach in Africa because this helps affect very greatly the spend, the way donor money is spent. We want to minimize the percentage of funds that are spent on anything other than medicines production itself. So we want to minimize the money that's spent on assuring quality, the minimum amount of money spent on managing, shipping, and procurement and testing. But another thing we do is we help assure the quality of medicines in Africa. The U.S. Pharmacopeal Drug Quality Initiative with Patrick Lucalet here in Washington took a survey in 2007 and found that 48% of the medicines that were imported into Africa that they sampled in 2007, they were either counterfeit or they were so substandard as to compromise drug quality. So we assist national drug regulatory agencies in Africa in reaching what's called strict regulatory authority status. There are companies who try to cheat. We want to help African countries throw out the companies that cheat by selling them counterfeit drugs and to decrease infrastructure cost, but something else is important. AIDS, tuberculosis, and malaria are not all of the diseases that people die from. Regional production enables Africa to make its own drugs for hypertension, for heart failure, for diabetes, for cancer, and for maternal and reproductive health. Well, the ACS GREET program has actually helped us quite a bit in doing this. And Chris King, who I think is probably listening, I hope is listening to this presentation, is someone who went with me to Africa last August to help teach at the Kilimanjaro School of Pharmacy, and we taught people how to make malaria drugs. There are about 300 million ACT treatments that are sourced globally by the Global Fund and the Affordable Medicines Facility for Malaria as of 2011. Typically, it now costs about 50 cents to $1.50 to manufacture pediatric and adult treatment regimens for malaria. The big challenge is to reduce these to about 10 cents per dose. That's effectively the cost of chloroquine as a single agent drug that needs to be completely replaced because of resistance. So we need to not only institute new green chemistry that can reduce the cost of manufacturing, but effectively to make the price of access approximate the cost of manufacturing. I'm going to speak to what we did in our GREET program in Africa. So there is a fixed dose combination ACT called dihydroartemisinin and piperaquine. Piperaquine phosphate is delivered as its tetraphosphate salt with 4 waters of hydration in the crystal. The piperaquine synthesis that we've come up with that is green approximates, and it has the same making and breaking of chemical bonds as earlier syntheses of piperaquine itself. 4,7-dichloroquinone is used, the starting material on the upper left-hand side. One of the chlorines is selectively replaced by piperazine. And then that molecule that you see in the right-hand side on the upper line is reacted with 1,3-dibromopropane, and 2 of those molecules come together to make the molecular structure of piperaquine as a free base. That's then converted into piperaquine phosphate salt. Dihydroartemisinin is derived really in one simple synthetic step from the natural product artemisinin itself. Sodium borohydride and calcium chloride are used to reduce the lactone ring, followed by the addition of aqueous hydrochloric acid so that the product crystallizes directly from the reaction to give dihydroartemisinin. And the stereochemistry of the alcohol and the lactone ring doesn't matter. But I'd like to draw your attention to this slide here. The new green process that we reproduced with our pharmaceutical partners in Africa reduces the E factor of making that simple molecule piperaquine from greater than 45 to about 7. And about 65% of that 7 kilos of waste that's generated for each kilo of drug can actually be recycled. This is the first time that drug was ever been synthesized in sub-Saharan Africa. 23 African pharmaceutical professionals synthesized both DHA from artemisinin, and the artemisinin came from an African company, and they made piperaquine. The starting materials for the piperaquine came from Sigma Aldrich in August of 2011 through the GREET program. After those participants made the drugs, the active pharmaceutical ingredients, they actually carried out a design of experiments and formulation equipment, and they made both pediatric and adult tablets of DHAP, that ACT that we're really interested in providing. Now, these are the people who did it. This is the pharmaceutical training class that we had that did that. Well, we've been very pleased at what we've been able to do in Africa. The United Nations has named us as a center of excellence in the African Initiative for New Drugs and Diagnostics. And we've worked with the Organization of African States to draw up a pharmaceutical manufacturing plan for Africa that has gone through, it's been presented to the Consolidated Ministers of Health, and it is momentarily, hopefully quickly, undergoing approval by the heads of state of the OAS so that we can work on a plan of funding widespread, quality-assured pharmaceutical manufacturing of essential medicines such as for malaria in Africa. This is something that has been built by the Kilimanjaro School of Pharmacy and the St. Luke Foundation. This is an actual small-scale pharmaceutical manufacturing pilot plant for making tablets. We can make about 50 kilo scale batches here. What I'd like to show you here, this is a blending machine, and so you're not so much interested in that because we're chemists, but what I want you to see is look at the reflection you can see in your floor. This is a high quality facility that would pass US FDA inspections. This is a tableting machine. This is where tablets of DHAP were made from the drugs that were made by our participants in Africa. This is just to show you what a granulation bowl looks like, where drugs are combined with inactive ingredients that assure their stability, their dissolution, their bioavailability, and bioequivalence so that drug performance can be reliable from batch to batch during production. These are some tablets of Efavirenz that we formulated and made in Africa during that same course. And here are some zebras. This is actually in the Serengeti Desert, and it's a very peaceful setting. It's a beautiful place. And this is somewhat less peaceful, but this is a good photograph. We came upon some lions that just brought down a Cape buffalo in the Serengeti. I'm sorry, no, this is from Ngorongoro Crater. And so I thought I'd show you this as well. This is the view that I can see from the St. Luke Foundation when I get up in the mornings to teach at the Kilimanjaro School of Pharmacy. And these are the people that I have to thank for an awful lot of the work that's going on here.
Thank you for watching this presentation. ACS Webinars is provided as a service of the American Chemical Society as your chemistry source for live weekly discussions and presentations that connect you with subject matter experts and global thought leaders concerning today's relevant professional issues in the chemical sciences, management, and business.